Speciation. Sexual selection can contribute to the formation of new species. When populations become reproductively isolated, different sexual selection pressures may act upon them, leading to the divergence of mating behaviors, preferences, and traits. Over time, this can result in the evolution of reproductive barriers and the formation of distinct species. Sexual dimorphism. Sexual selection often leads to pronounced differences between males and females of the same species, known as sexual dimorphism. Males may develop elaborate features or behaviors to attract females, while females may possess traits that enhance their ability to choose high-quality mates or provide parental care. Examples include the contrasting size and coloration between male and female birds or the elaborate displays of male fish during courtship. Coevolution of traits. Selection can drive the coevolution of traits in males and females. For instance, as males develop more intricate courtship displays or mating behaviors, females may evolve sensory adaptations to better evaluate and respond to these displays. This coevolutionary process can result in an ongoing arms race between the sexes, leading to the diversification and refinement of mating strategies. Impact on genetic variation. Sexual selection can influence the genetic variation within populations. Traits under strong sexual selection pressure may experience rapid evolutionary changes, leading to high levels of genetic diversity. Additionally, sexual selection can contribute to the maintenance of genetic variation by promoting the maintenance of different alleles associated with mating success. Social structures and behaviors. Sexual selection can influence the development of social structures and behaviors within species. In species with intense intersexual competition, hierarchical systems or complex social interactions may arise. Additionally, the presence of intersexual selection can lead to the evolution of cooperative behaviors or elaborate courtship rituals. It is important to recognize that sexual selection is not the sole driving force of evolution. It works in conjunction with other evolutionary mechanisms, such as natural selection, genetic drift, and gene flow, to shape the diversity and adaptations observed in the animal kingdom. Furthermore, the intensity and direction of sexual selection can vary among species, populations, and ecological contexts. Overall, sexual selection, through intersexual and intersexual selection, plays a vital role in shaping the evolutionary trajectories of species. It drives the evolution of sexually dimorphic traits, influences reproductive strategies, and contributes to the formation of new species. By understanding the mechanisms and implications of sexual selection, we can gain insights into the complexity of animal behavior, reproduction, and the diversity of life on Earth. Secondary sexual characteristics. Sexy Sun hypothesis. Good gene hypothesis in evolution in detail. Big essay with related to zoology. ChatGPT. Secondary sexual characteristics and their role in evolution. Introduction. Secondary sexual characteristics are traits that differ between males and females of the same species and are involved in mate attraction, mate choice, and competition for mates. These characteristics play a crucial role in the process of sexual selection, a specific form of natural selection that operates on traits related to reproduction. In the field of zoology, secondary sexual characteristics have been extensively studied to understand their evolutionary significance and the mechanisms driving their development. Two well-known hypotheses related to secondary sexual characteristics are the Sexy Sun hypothesis and the Good Genes hypothesis. These hypotheses provide insights into the evolutionary processes underlying the development and maintenance of secondary sexual characteristics. The Sexy Sun hypothesis. The Sexy Sun hypothesis, proposed by Ronald Fisher, suggests that females choose mates based on traits that will enhance the reproductive success of their male offspring. According to this hypothesis, females prefer males with attractive or conspicuous traits because these traits will be inherited by their sons, making them more successful in attracting mates in future generations. By selecting mates with such traits, females indirectly increase the reproductive success of their offspring. For example, in many bird species, males exhibit elaborate plumage or vocal displays. Females are more likely to choose males with exaggerated traits because they perceive them as attractive and of high genetic quality. The offspring of these attractive males will inherit the same attractive traits, increasing their chances of successful reproduction. This preference for attractive traits by females contributes to the evolutionary maintenance and exaggeration of secondary sexual characteristics. The Good Genes hypothesis. The Good Genes hypothesis proposes that females choose mates based on traits that indicate good genetic quality and fitness. According to this hypothesis, the preferred traits are indicators of the male's ability to survive, resist diseases, or produce healthy offspring. By selecting mates with these traits, females increase the genetic quality of their offspring, leading to improved survival and reproductive success. One example of the Good Genes hypothesis is the choice of females for males with bright coloration in fish species. Bright coloration is often associated with high levels of antioxidants, which can indicate the male's ability to resist oxidative stress and maintain good health. Females prefer males with such traits because they provide genetic benefits to the offspring, including improved resistance to diseases and increased survival chances. Advantages and disadvantages of secondary sexual characteristics. Secondary sexual characteristics provide several advantages to individuals in the context of reproduction and evolutionary fitness. These advantages include increased attractiveness. Secondary sexual characteristics make individuals more attractive to potential mates, increasing their chances of successful reproduction. Competitive advantage. In species with intense male-male competition, secondary sexual characteristics can provide a competitive edge in acquiring mates and securing breeding territories. Honest signaling. Secondary sexual characteristics often signal the overall health, genetic quality, and fitness of individuals. By choosing mates with attractive traits, females can ensure the acquisition of good genes for their offspring. Despite these advantages, secondary sexual characteristics also have some drawbacks, including energy and resource costs. Developing and maintaining elaborate secondary sexual characteristics can require significant energy and resources, diverting them from other important functions such as growth, survival, or parental care. Predation risk. Exaggerated secondary sexual characteristics can make individuals more visible to predators, increasing their vulnerability to predation. Intersexual conflict. Secondary sexual characteristics can lead to conflicts between males and females. For example, males may coerce females into mating, leading to negative fitness consequences for females. Application in zoology. The study of secondary sexual characteristics and their role in evolution has important implications for understanding reproductive strategies, mate choice, and sexual dimorphism in various animal species. Researchers in zoology use these concepts to investigate topics such as species diversification, sexual selection, and the evolution of mating behaviors. For example, studies. Continuation. ChatGPT. Have shown that secondary sexual characteristics can drive speciation by promoting reproductive isolation between populations. Differences in these traits can lead to mate preferences and mate choice divergence, ultimately resulting in the formation of new species. Additionally, the study of secondary sexual characteristics helps researchers understand the mechanisms underlying mate choice and sexual selection. By examining the preferences of females and the traits favored by selection, scientists can gain insights into the evolutionary pressures shaping the development and maintenance of these characteristics. This knowledge contributes to our understanding of the processes driving the evolution of sexual dimorphism and the diversity of mating systems across different animal taxa. Furthermore, the study of secondary sexual characteristics has practical applications in conservation biology. Some species rely on specific secondary sexual traits for courtship and mating rituals, and the loss or degradation of these traits can have detrimental effects on population viability. By understanding the importance of these traits in reproductive success, conservation efforts can be directed towards preserving and restoring habitats that support the expression of secondary sexual characteristics, thereby ensuring the long-term survival of species. In conclusion, secondary sexual characteristics play a crucial role in evolution, particularly in the context of mate attraction, mate choice, and sexual selection. The sexy son hypothesis and the good genes hypothesis provide theoretical frameworks to explain the evolutionary significance of these traits. While secondary sexual characteristics confer advantages such as increased attractiveness and reproductive success, they also come with costs and potential conflicts. The study of these characteristics in zoology has broad applications, ranging from understanding species diversification to informing conservation strategies. By unraveling the complexities of secondary sexual characteristics, researchers can deepen our understanding of the fascinating evolutionary processes that shape the diversity of life on Earth. Population genetics. Populations, gene pool in detail with respect to evolution with examples of zoology in detail. ChatGPT. Population genetics is a branch of biology that studies the genetic composition and changes within populations over time. It provides insights into the mechanisms of evolution and how genetic variation is inherited and distributed among individuals within a population. Understanding population genetics is essential for studying the processes that drive evolutionary change, including natural selection, genetic drift, gene flow, and mutation. With respect to zoology, population genetics helps us understand how populations of animals evolve and adapt to their environments. A population is a group of individuals of the same species that live in the same geographic area and have the potential to interbreed. The gene pool of a population refers to all the alleles present in the population for a particular gene or set of genes. It represents the total genetic diversity within the population. One of the key concepts in population genetics is allele frequency, which refers to the relative abundance of a particular allele in a population. Allele frequencies can change over time due to various evolutionary forces. Let's explore these forces and their impact on the gene pool using examples from zoology. Natural selection. Natural selection is the process by which certain traits become more or less common in a population based on their fitness. For example, in a population of birds, those with longer beaks may be better adapted for extracting nectar from flowers, leading to higher reproductive success. Over time, the frequency of the allele for long beaks may increase in the population, resulting in evolutionary change. Genetic drift. Genetic drift refers to random fluctuations in allele frequencies in small populations. It can have a significant impact on genetic diversity, especially in isolated populations. For instance, in a small population of lizards on an isolated island, a particular allele may become more or less common by chance rather than by natural selection. Gene flow. Gene flow occurs when individuals migrate between populations, resulting in the transfer of genetic material. This can introduce new alleles into a population or alter the frequency of existing alleles. For example, if a group of birds migrates from one habitat to another and mates with the local population, their genetic contribution can influence the allele frequencies in the recipient population. Mutation. Mutation is the ultimate source of genetic variation. New mutations can introduce novel alleles into a population. While most mutations are neutral or detrimental, some may confer advantageous traits that increase an individual's fitness. Over time, these beneficial mutations can become more common in a population through natural selection. By studying population genetics, researchers can analyze genetic data to estimate population sizes, track changes in allele frequencies, and infer the evolutionary history of populations. This information is crucial for understanding species adaptations to their environments, the potential for speciation, and the conservation of endangered species. Additionally, population genetics can provide insights into disease susceptibility, genetic disorders, and human evolution. In conclusion, population genetics is a powerful tool for understanding how genetic variation is distributed and changes within populations over time. It helps us unravel the mechanisms of evolution, including natural selection, genetic drift, gene flow, and mutation. By studying the gene pool of populations, researchers can gain insights into the evolutionary processes that shape the diversity of animal species, and apply this knowledge to various fields, such as conservation biology and human health. Gene frequency, in detail with respect to evolution with examples of zoology in detail. ChatGPT. Gene frequency, also known as allele frequency, refers to the proportion of a particular allele within a population. It represents the relative abundance of a specific variant of a gene within the gene pool of a population. Understanding gene frequency is crucial in studying the patterns of genetic variation and how they change over time in the context of evolution. Here's a detailed explanation of gene frequency with examples from zoology. Hardy-Weinberg equilibrium. The Hardy-Weinberg equilibrium is a fundamental principle in population genetics that describes the relationship between allele frequencies and genotype frequencies in an idealized population. According to this principle, in the absence of evolutionary forces such as natural selection, genetic drift, gene flow, and mutation, the allele frequencies within a population will remain constant from one generation to the next. This equilibrium provides a baseline against which changes in gene frequencies can be measured. Genetic drift. Genetic drift refers to random fluctuations in allele frequencies within a population due to chance events. It is especially pronounced in small populations. For example, in a small population of birds, genetic drift may lead to the loss of certain alleles over time. If the individuals carrying a particular allele do not reproduce or their offspring do not survive, the frequency of that allele will decrease, potentially leading to its eventual disappearance from the population. Natural selection. Natural selection is a powerful force that can influence gene frequencies within a population. Certain alleles may confer advantages or disadvantages to individuals in terms of survival and reproduction. As a result, alleles that enhance fitness tend to increase in frequency, while those that reduce fitness tend to decrease. For instance, in a population of gazelles, individuals with alleles that enable faster running speeds may have a higher chance of escaping predators and reproducing, leading to an increase in the frequency of those alleles over time. Gene flow. Gene flow refers to the movement of genetic material from one population to another through migration and mating. When individuals migrate between populations and successfully reproduce, they introduce their alleles into the new population, potentially altering the gene frequencies. For example, if a population of frogs inhabiting a particular region expands its range and mates with a geographically isolated population, gene flow can increase the genetic diversity and alter the gene frequencies in both populations. Mutation. Mutations are the ultimate source of genetic variation. They introduce new alleles into a population, thereby potentially altering gene frequencies. While most mutations are neutral or detrimental, some may confer advantageous traits that increase an individual's fitness. If these beneficial alleles provide a selective advantage, they are likely to increase in frequency over time through natural selection. An example is the development of antibiotic resistance in bacteria, where mutations in certain genes allow bacteria to survive and reproduce in the presence of antibiotics. Studying gene frequencies provides valuable insights into the genetic structure of populations and the mechanisms driving evolutionary change. By analyzing changes in gene frequencies over time, researchers can infer the effects of evolutionary forces such as natural selection, genetic drift, gene flow, and mutation. This information is crucial for understanding the genetic basis of traits, speciation processes, population dynamics, and the conservation of endangered species. Additionally, gene frequency data can be used to investigate human evolution, genetic disorders, and the spread of diseases. In conclusion, gene frequency is a key concept in population genetics and provides important information about the genetic variation within populations and how it changes over time. By studying gene frequencies in zoology, researchers can gain insights into the mechanisms of evolution, track the effects of evolutionary forces, and understand the genetic basis of traits in animal populations. Hardy-Weinberg. Law in detail with respect to evolution with examples of zoology in detail. ChatGPT. The Hardy-Weinberg law, also known as the Hardy-Weinberg equilibrium, is a fundamental principle in population genetics that describes the relationship between allele frequencies and genotype frequencies in an idealized population. It provides a mathematical framework to understand how genetic variation is maintained within populations over time. The Hardy-Weinberg law is based on several assumptions, including a large population size, random mating, no mutation, no migration, and no natural selection. Here's a detailed explanation of the Hardy-Weinberg law and its application in zoology. Hardy-Weinberg equilibrium equation. The Hardy-Weinberg law is expressed as an equation, p squared plus 2pq plus q squared equals 1, where p represents the frequency of one allele A and q represents the frequency of the other allele A in a population. The equation describes the proportions of different genotypes in the population, homozygous dominant AA, heterozygous A, and homozygous recessive A. According to the Hardy-Weinberg equilibrium, these proportions remain constant from one generation to the next. Assumptions of the Hardy-Weinberg law. The Hardy-Weinberg law assumes specific conditions within a population. Large population size. The population size is large enough to prevent random fluctuations in allele frequencies due to genetic drift. Random mating. Individuals in the population mate randomly, without any preference for particular genotypes. No mutation. There are no new mutations introducing new alleles into the population. No migration. There is no migration of individuals in or out of the population, which would introduce new alleles or remove existing ones. No natural selection. There is no differential survival or reproductive advantage associated with specific genotypes. Application in zoology. The Hardy-Weinberg law is a useful tool for studying genetic variation in animal populations and understanding evolutionary processes. It allows researchers to determine whether a population is in equilibrium and to infer the presence of evolutionary forces acting on the population. Deviations from the Hardy-Weinberg equilibrium indicate that one or more of the assumptions are violated, suggesting the influence of evolutionary processes. Examples of the Hardy-Weinberg law in zoology include coat color in mammals. In a population of rabbits, coat color is determined by a single gene with two alleles, brown B and white B. By analyzing the frequencies of the brown and white alleles in the population, researchers can predict the expected proportions of different coat color genotypes under the Hardy-Weinberg equilibrium. Blood types in humans. The ABO blood type system is governed by multiple alleles. By examining the frequencies of the A, B, and O alleles in a human population, researchers can use the Hardy-Weinberg equation to predict the expected proportions of different blood types. Significance. The Hardy-Weinberg law serves as a null model for understanding the dynamics of genetic variation in populations. If a population deviates from the Hardy-Weinberg equilibrium, it suggests that evolutionary processes are at play, such as natural selection, genetic drift, mutation, migration, or non-random mating. Studying deviations from equilibrium can provide insights into the factors driving genetic changes in populations and the mechanisms of evolution. In conclusion, the Hardy-Weinberg law is a fundamental principle in population genetics that describes the relationship between allele frequencies and genotype frequencies in an idealized population. It provides a baseline against which changes in genetic variation can be measured and allows researchers to infer the presence of evolutionary forces. By applying the Hardy-Weinberg equation, zoologists can study genetic variation in animal populations and gain insights into Founder principle, in detail with respect to evolution with examples of zoology in detail. ChatGPT. The founder principle is a concept in population genetics that explains how genetic variation can be influenced when a small group of individuals establishes a new population. It occurs when a few individuals, known as founders, migrate or become isolated from a larger population and establish a new colony in a different geographic area. The founder principle has significant implications for evolutionary processes and can lead to unique genetic characteristics in the newly formed population. Here's a detailed explanation of the founder principle and its application in zoology. Establishment of a new population. When a small group of individuals becomes isolated or migrates to a new habitat, they establish a new population. This founding population represents a subset of the original population and carries a limited amount of genetic variation. Genetic drift. Genetic drift, the random fluctuation of allele frequencies, plays a crucial role in the founder principle. In small founder populations, genetic drift can have a pronounced effect on the gene pool due to the limited number of individuals. Random events and chance occurrences can cause certain alleles to become more or less prevalent in subsequent generations. Loss of genetic diversity. Due to the small size of the founding population, genetic diversity is reduced compared to the original population. Some alleles may be lost entirely, while others may become fixed, meaning they are the only variant present in the population. Genetic bottleneck. The founder principle is closely related to the concept of a genetic bottleneck. A genetic bottleneck occurs when a large population undergoes a significant reduction in size, leading to a small number of individuals founding a new population. This process can lead to the loss of genetic variation and an increased likelihood of genetic diseases or abnormalities. Founder effects. The founder principle results in what is known as a founder effect, where the genetic composition of the new population is influenced by the alleles carried by the founders. Rare or uncommon alleles in the original population may become more prevalent or even fixed in the founder population. This can result in unique genetic characteristics that are distinct from the original population. Examples of the founder principle in zoology include Darwin's finches. In the Galapagos Islands, a group of finches migrated from the mainland and established different populations on different islands. Each population faced unique environmental conditions and selective pressures, leading to the evolution of different beak shapes and sizes among the various species of Darwin's finches. Island populations. Many island ecosystems are the result of founder events, where a few individuals colonized an island and gave rise to a new population. These populations often exhibit unique adaptations and genetic characteristics due to their isolated and restricted environments. Lake Victoria cichlids. The cichlid fish species in Lake Victoria experienced a founder event when the lake formed. A small number of individuals colonized the lake and gave rise to a diverse array of cichlid species with different coloration, body shapes, and feeding habits. The founder principle demonstrates how the establishment of new populations through migration or isolation can significantly impact genetic variation. The reduced genetic diversity and founder effects can lead to the development of unique traits and adaptations in populations, ultimately contributing to evolutionary change and diversification in zoology. Bottleneck effect and genetic drift as factors in evolution in detail with respect to evolution with examples of zoology in detail. ChatGPT. The bottleneck effect and genetic drift are important factors in evolution that can significantly impact the genetic diversity and composition of populations. They play a crucial role in shaping the evolutionary trajectory of species. Let's explore these concepts in detail and their relevance to zoology. Bottleneck effect. The bottleneck effect occurs when a large population experiences a drastic reduction in size due to a catastrophic event, such as natural disasters, disease outbreaks, or human-induced factors. The surviving individuals become the founders of a new population, resulting in a significant loss of genetic variation. Here are some key points. Genetic diversity reduction. The bottleneck effect leads to a decrease in the number of alleles and genotypes in the population. Some alleles may be lost entirely, while others become overrepresented. Increased genetic drift. With a smaller population size, genetic drift becomes more influential. Random fluctuations in allele frequencies can have a greater impact, leading to rapid changes in the genetic makeup of the population. Loss of adaptive traits. If a bottleneck event selectively eliminates individuals with specific traits, important adaptive traits may be lost. This can reduce the population's ability to respond to environmental changes and increase its vulnerability to further challenges. Example in zoology. The northern elephant seal is a classic example of the bottleneck effect. In the 19th century, their population was reduced to a few dozen individuals due to excessive hunting. As a result, the genetic diversity of the population decreased significantly. Today, the population has rebounded but retains low genetic variation compared to their original numbers. Genetic drift. Genetic drift refers to random fluctuations in allele frequencies within a population over time. It occurs due to chance events rather than natural selection. Genetic drift is more pronounced in small populations and can lead to significant changes in the genetic composition. Here are some key points. Random sampling. In small populations, the allele frequencies in subsequent generations can deviate from the original frequencies due to chance alone. Certain alleles may Founder principle, in detail with respect to evolution with examples of zoology in detail. ChatGPT. The founder principle is a concept in population genetics that explains how genetic variation can be influenced when a small group of individuals establishes a new population. It occurs when a few individuals, known as founders, migrate or become isolated from a larger population and establish a new colony in a different geographic area. The founder principle has significant implications for evolutionary processes and can lead to unique genetic characteristics in the newly formed population. Here's a detailed explanation of the founder principle and its application in zoology. Establishment of a new population. When a small group of individuals becomes isolated or migrates to a new habitat, they establish a new population. This founding population represents a subset of the original population and carries a limited amount of genetic variation. Genetic drift. Genetic drift, the random fluctuation of allele frequencies, plays a crucial role in the founder principle. In small founder populations, genetic drift can have a pronounced effect on the gene pool due to the limited number of individuals. Random events and chance occurrences can cause certain alleles to become more or less prevalent in subsequent generations. Loss of genetic diversity. Due to the small size of the founding population, genetic diversity is reduced compared to the original population. Some alleles may be lost entirely, while others may become fixed, meaning they are the only variant present in the population. Genetic bottleneck. The founder principle is closely related to the concept of a genetic bottleneck. A genetic bottleneck occurs when a large population undergoes a significant reduction in size, leading to a small number of individuals founding a new population. This process can lead to the loss of genetic variation and an increased likelihood of genetic diseases or abnormalities. Founder effects. The founder principle results in what is known as a founder effect, where the genetic composition of the new population is influenced by the alleles carried by the founders. Rare or uncommon alleles in the original population may become more prevalent or even fixed in the founder population. This can result in unique genetic characteristics that are distinct from the original population. Examples of the founder principle in zoology include Darwin's finches. In the Galapagos Islands, a group of finches migrated from the mainland and established different populations on different islands. Each population faced unique environmental conditions and selective pressures, leading to the evolution of different beak shapes and sizes among the various species of Darwin's finches. Island populations. Many island ecosystems are the result of founder events, where a few individuals colonized an island and gave rise to a new population. These populations often exhibit unique adaptations and genetic characteristics due to their isolated and restricted environments. Lake Victoria cichlids. The cichlid fish species in Lake Victoria experienced a founder event when the lake formed. A small number of individuals colonized the lake and gave rise to a diverse array of cichlid species with different coloration, body shapes, and feeding habits. The founder principle demonstrates how the establishment of new populations through migration or isolation can significantly impact genetic variation. The reduced genetic diversity and founder effects can lead to the development of unique traits and adaptations in populations, ultimately contributing to evolutionary change and diversification in zoology. Bottleneck effect and genetic drift as factors in evolution in detail with respect to evolution with examples of zoology in detail. ChatGPT. The bottleneck effect and genetic drift are important factors in evolution that can significantly impact the genetic diversity and composition of populations. They play a crucial role in shaping the evolutionary trajectory of species. Let's explore these concepts in detail and their relevance to zoology. Bottleneck effect. The bottleneck effect occurs when a large population experiences a drastic reduction in size due to a catastrophic event, such as natural disasters, disease outbreaks, or human-induced factors. The surviving individuals become the founders of a new population, resulting in a significant loss of genetic variation. Here are some key points. Genetic diversity reduction. The bottleneck effect leads to a decrease in the number of alleles and genotypes in the population. Some alleles may be lost entirely, while others become overrepresented. Increased genetic drift. With a smaller population size, genetic drift becomes more influential. Random fluctuations in allele frequencies can have a greater impact. Random fluctuations in allele frequencies can have a greater impact, leading to rapid changes in the genetic makeup of the population. Adaptive traits. If a bottleneck event selectively eliminates individuals with specific traits, important adaptive traits may be lost. This can reduce the population's ability to respond to environmental changes and increase its vulnerability to further challenges. Example in zoology. The northern elephant seal is a classic example of a bottleneck effect. In the 19th century, their population was reduced to a few dozen individuals due to excessive hunting. As a result, the genetic diversity of the population decreased significantly. Today, the population has rebounded but retains low genetic variation compared to their original numbers. Genetic drift. Genetic drift refers to random fluctuations in allele frequencies within a population over time. It occurs due to chance events rather than natural selection. Genetic drift is more pronounced in small populations and can lead to significant changes in the genetic composition. Here are some key points. Random sampling. In small populations, the allele frequencies in subsequent generations can deviate from the original frequencies due to chance alone. Certain alleles may become more or less common over time, leading to the loss or fixation of alleles. Founder effect. Genetic drift is closely related to the founder effect, which occurs when a small group establishes a new population. The limited gene pool of the founders can lead to a different genetic composition in the new population compared to the source population. Increased inbreeding. Genetic drift can increase the likelihood of inbreeding in small populations. Inbreeding reduces genetic diversity and increases the expression of harmful recessive traits, potentially compromising the fitness of the population. Example in zoology. The Florida panther population in the United States experienced severe genetic drift due to a small founding population and subsequent isolation. This led to increased inbreeding and reduced genetic diversity, resulting in various health issues and reduced reproductive success in the population. Both the bottleneck effect and genetic drift highlight the significant impact of random events and population size on the genetic makeup of populations. Understanding these factors is essential in studying the evolutionary processes and conservation strategies for species in zoology. Evidence for evolution. DNA evidence, in detail with respect to evolution with examples of zoology in detail. ChatGPT. DNA evidence is a powerful tool that provides compelling evidence for evolution. It allows scientists to study genetic similarities and differences among species, providing insights into their evolutionary relationships and patterns of descent. Here's a detailed explanation of DNA evidence in the context of evolution, along with examples from zoology. DNA sequencing and phylogenetics. DNA sequencing technology has revolutionized our understanding of evolution. By comparing DNA sequences from different organisms, scientists can construct phylogenetic trees that depict the evolutionary relationships between species. Here are some key points. Homologous genes. DNA sequences can be compared to identify homologous genes, which are genes inherited from a common ancestor. The degree of similarity in DNA sequences reflects the degree of relatedness between species. Molecular clock. DNA sequences can be used to estimate the time since two species diverged from a common ancestor. Mutations occur at a relatively constant rate, providing a molecular clock for estimating evolutionary divergence. Cladistics. DNA data are often used in cladistic analyses, where species are grouped based on shared derived characters. DNA sequences provide valuable characters for inferring evolutionary relationships and constructing cladograms. Example in zoology. DNA evidence has played a crucial role in understanding the evolutionary relationships among primates. By comparing DNA sequences from humans, chimpanzees, gorillas, and other primate species, scientists have confirmed that humans share a more recent common ancestor with chimpanzees than with other primates. This finding supports the theory of common descent. Comparative genomics. Comparative genomics involves comparing the entire genomes of different organisms to identify similarities and differences. It allows researchers to explore genomic changes that have occurred during evolutionary history. Here are some key points. Genomic rearrangements. Comparative genomics reveals large-scale genomic rearrangements, such as inversions, duplications, and translocations. These rearrangements can provide insights into evolutionary events and the divergence of species. Gene families and duplication. Comparative genomics helps identify gene families, which are groups of genes derived from a common ancestral gene through duplication events. Studying gene families can provide insights into the evolution of new functions and adaptations. Pseudogenes and non-coding DNA. Comparative genomics also reveals the presence of pseudogenes and non-coding DNA regions. Pseudogenes are non-functional copies of genes that have accumulated mutations over time. Non-coding DNA regions can provide clues about regulatory elements and evolutionary changes. Example in zoology. Comparative genomics has shed light on the evolution of whales. By comparing the genomes of whales with those of their terrestrial relatives, scientists have discovered genetic evidence for the transition from land to water. They identify genes associated with marine adaptations, such as changes in limb structure and the loss of certain olfactory genes. DNA evidence is a cornerstone of modern evolutionary biology. It provides detailed insights into the evolutionary history of organisms, helps reconstruct phylogenetic relationships, and contributes to our understanding of the mechanisms driving evolutionary change. Through DNA sequencing and comparative genomics, scientists can unravel the genetic basis of evolution and the interconnectedness of species in the animal kingdom. Evolution fossil evidence, in detail with respect to evolution with examples of zoology in detail. ChatGPT. Fossil evidence is one of the most important lines of evidence for understanding evolution. Fossils are the preserved remains or traces of organisms from the past, providing a unique window into the history of life on Earth. They offer valuable insights into the process of evolution, the origin of new species, and the patterns of biodiversity over time. Here's a detailed explanation of fossil evidence in the context of evolution, along with examples from zoology. Transitional fossils. Transitional fossils are fossils that exhibit characteristics intermediate between two major groups of organisms. They provide direct evidence of evolutionary links between different species or lineages. These fossils help fill in the gaps in the fossil record and show the gradual changes that occurred during the evolution of new traits or the origin of new species. Example in zoology. One well-known example of a transitional fossil is Archaeopteryx, an ancient bird-like dinosaur that lived around 150 million years ago. Archaeopteryx exhibits features of both reptiles and birds, such as feathers, wings, and reptilian characteristics like teeth and a long bony tail. It represents an intermediate stage between non-avian dinosaurs and modern birds, providing evidence for the evolution of flight. Succession of fossils. The study of fossil assemblages in different rock layers reveals a pattern of succession, where simpler organisms appear in older layers while more complex organisms appear in more recent layers. This pattern supports the idea of gradual changes in species over time, with new forms evolving from earlier forms. Example in zoology, the fossil record of marine invertebrates, such as trilobites, ammonites, and brachiopods, shows a clear succession of different groups of organisms through time. Simple marine organisms like trilobites appear in the early Paleozoic era, while more complex forms like ammonites dominate the Mesozoic era. This succession of fossils demonstrates the progressive diversification and evolution of marine life over millions of years. Extinction events. Fossils also provide evidence of mass extinction events, where a significant number of species become extinct within a relatively short period. These events have had a profound impact on the course of evolution by eliminating certain lineages and creating opportunities for new ones to emerge. Example in zoology, the Cretaceous-Paleogene extinction event, which occurred around 66 million years ago, led to the extinction of non-avian dinosaurs, as well as many other species. Fossils of dinosaurs in upper Cretaceous sediments abruptly disappear above a layer rich in the element iridium, indicating a catastrophic event such as an asteroid impact. This extinction event opened up ecological niches, allowing mammals and birds to diversify and become the dominant terrestrial vertebrates. Biogeography. Fossils found in different geographical locations provide insights into the distribution and migration of organisms over time. They help us understand how species have spread and adapted to different environments, providing evidence for evolutionary processes such as speciation and adaptive radiation. Example in zoology, fossil evidence of marsupials in South America and Australia suggests that these two continents were once connected. The presence of similar marsupial groups in both regions indicates that they shared a common ancestor before the continents drifted apart. The distribution of fossils supports the hypothesis of continental drift and provides insights into the evolutionary history of marsupials. Fossil evidence plays a crucial role in documenting the history of life on Earth and supporting the theory of evolution. By studying fossils, scientists can reconstruct evolutionary lineages, identify key transitional forms, and understand the processes that have shaped biodiversity over millions of years. Fossil evidence, combined with other lines of evidence such as DNA analysis, helps paint a comprehensive picture of the evolutionary history of organisms, including the diverse array of species in the evolution embryological. Evidence, in detail with respect to evolution with examples of zoology in detail. ChatGPT. Embryological evidence is another line of evidence that supports the theory of evolution. It involves the study of the developmental stages of organisms from fertilization to the formation of the adult form. Embryological similarities among different species provide insights into their shared evolutionary history and the patterns of common ancestry. Here's a detailed explanation of embryological evidence in the context of evolution, along with examples from zoology. Embryonic developmental stages. Comparative embryology involves comparing the developmental stages of embryos across different species. This allows scientists to identify similarities and differences in the early stages of development and uncover patterns of evolutionary relationships. Example in zoology, the embryonic development of vertebrates, such as fish, amphibians, reptiles, birds, and mammals, shows striking similarities during the early stages. For instance, all vertebrate embryos possess a notochord, pharyngeal pouches, and a tail during certain stages of development. These shared features suggest a common ancestry and support the idea of evolution from a common ancestor. Homologous structures. Embryological evidence also involves the study of homologous structures, which are structures that have a similar origin and development but may have different functions in different species. These structures provide evidence for shared ancestry and evolutionary relationships. Example in zoology, the forelimbs of vertebrates, such as humans, bats, whales, and cats, exhibit homologous structures despite having different functions. They all share a similar pattern of bones, including the humerus, radius, and ulna. This similarity suggests that these species share a common ancestor with forelimbs adapted for different purposes through evolutionary modification. Developmental gene expression. Embryological evidence also includes the study of gene expression during development. By comparing the expression of genes across different species, scientists can identify shared genetic pathways and regulatory mechanisms that control development. Example in zoology, the Hox genes play a crucial role in embryonic development and determine the body plan of organisms. These genes are highly conserved across a wide range of animal species. By studying the expression patterns of Hox genes during embryonic development, scientists have identified similarities in the arrangement and function of body segments across different species. This supports the idea of shared ancestry and evolutionary relationships. Vestigial structures. Embryological evidence can also be seen in the presence of vestigial structures during development. Vestigial structures are remnants of functional organs or structures that were more developed in ancestral species but have reduced or no function in the current species. Example in zoology, the presence of a small, non-functional tailbone coccyx in humans is considered a vestigial structure. During embryonic development, humans possess a tail-like structure that later regresses, leaving behind the coccyx. This indicates a shared developmental pattern with other vertebrates that have functional tails. The presence of vestigial structures in embryonic development provides evidence of evolutionary history. Embryological evidence helps uncover patterns of evolutionary relationships and provides insights into the shared ancestry of organisms. By comparing the developmental stages, structures, gene expression, and presence of vestigial structures, scientists can reconstruct the evolutionary history of species and understand how they have diversified over time. Embryological evidence, combined with other lines of evidence, contributes to our understanding of the evolutionary process and the interconnectedness of all living organisms. Evolution geological evidence, in detail with respect to evolution with examples of zoology in detail. ChatGPT. Geological evidence, also known as paleontological evidence, is a crucial line of evidence that supports the theory of evolution. It involves the study of fossils in the geological record to understand the history of life on Earth and the changes that have occurred over time. Here's a detailed explanation of geological evidence in the context of evolution, along with examples from zoology. Fossil record. The fossil record provides direct evidence of past life forms and their characteristics. Fossils are the preserved remains or traces of once living organisms, and they can be found in various forms, including bones, shells, teeth, imprints, and tracks. By examining fossils, scientists can determine the existence of different species and their evolutionary relationships. Example in zoology, the discovery of fossilized dinosaur bones has provided extensive evidence of the existence and evolution of dinosaurs. Fossils of various dinosaur species, such as Tyrannosaurus rex, Triceratops, and Stegosaurus, have been found in different geological layers, showing the diversity and evolution of dinosaurs over millions of years. Transitional fossils. Transitional fossils are fossils that exhibit intermediate characteristics between different species, providing evidence of evolutionary transitions. These fossils are crucial in documenting the gradual changes that occurred during the evolution of one species into another. Example in zoology, the fossil record includes several transitional fossils that provide evidence of evolutionary transitions. One famous example is the fossil of Archaeopteryx, which exhibits features of both reptiles and birds. It has reptilian characteristics like teeth and a long tail, along with bird-like features such as feathers and wings. This fossil provides evidence for the evolutionary link between dinosaurs and birds. Geological strata and dating methods. Geological strata, or layers of rock, provide a chronological record of Earth's history. By studying the sequence and relative positions of different rock layers, scientists can determine the relative age of fossils and the order in which organisms live. Radiometric dating methods, such as carbon dating and potassium argon dating, are used to assign absolute ages to fossils and rock layers. Example in zoology, the Burgess Shale in Canada is a famous fossil site known for its exceptional preservation of soft-bodied organisms from the Cambrian period. The fossils found in this geological formation provide valuable insights into the early diversification of animal life on Earth and the emergence of complex body plans. Biogeography. Biogeography, the study of the geographic distribution of organisms, also provides geological evidence for evolution. The distribution patterns of species across different continents, islands, and habitats can be explained by their evolutionary history and the movement of land masses over time. Example in zoology, the distribution of marsupials, such as kangaroos, in Australia and their absence in other parts of the world as a result of their unique evolutionary history. The separation of Australia from other continents allowed marsupials to evolve and diversify in isolation, leading to their exclusive presence in Australia. Geological evidence plays a vital role in understanding the history of life on Earth and the processes that have shaped its diversity. By studying fossils, geological strata, transitional forms, and biogeographical patterns, scientists can reconstruct the evolutionary timeline, identify ancestral relationships, and trace the changes that have occurred over millions of years. The geological record provides tangible evidence for the theory of evolution and contributes to our understanding of the interconnectedness and dynamic nature of life on our planet. Evolution in action in detail. Essay in detail with respect to evolution with examples of zoology in detail. ChatGPT. Evolution in action refers to the ongoing process of genetic change and adaptation in populations over time. It provides evidence for the theory of evolution by demonstrating how species can change and diversify in response to environmental pressures. Here's a detailed essay on evolution in action, with examples from zoology. Evolution in action, examples from zoology. Antibiotic resistance in bacteria. One of the most well-known examples of evolution in action is the development of antibiotic resistance in bacteria. When bacteria are exposed to antibiotics, some individuals may possess genetic variations that make them resistant to the drug. These resistant bacteria have a selective advantage, as they can survive and reproduce while non-resistant individuals die. Over time, the population becomes dominated by antibiotic-resistant bacteria, reducing the effectiveness of the antibiotic. Example in zoology, methicillin-resistant Staphylococcus aureus MRSA is a bacterium that has evolved resistance to multiple antibiotics, including methicillin. This evolution of antibiotic resistance poses a significant challenge in the field of medicine, as it requires the development of new antibiotics to combat these resistant strains. Industrial melanism in moths. Industrial melanism is a phenomenon observed in some moth populations living in industrial areas with high pollution levels. Initially, these moths had light-colored wings, which provided camouflage against tree trunks. However, with the advent of industrial pollution and the darkening of tree trunks due to soot deposits, darker variants of moths became more advantageous as they were better camouflaged against the new background. Over time, the proportion of dark-colored moths increased due to natural selection. Example in zoology, the peppered moth Piston beetleria is a classic example of industrial melanism. In areas affected by industrial pollution during the Industrial Revolution, the dark-colored melanic form of the moth became more prevalent, while the light-colored form declined. As pollution levels have decreased in recent years, the light-colored form has seen a resurgence in populations. Beak adaptations in Darwin's finches. The finches of the Galapagos Islands, famously studied by Charles Darwin, exhibit remarkable beak variations that have evolved in response to different food sources. The availability of various food types on the islands has exerted selective pressure on the finches, leading to the development of different beak shapes and sizes that are specialized for specific diets. This is a classic example of adaptive radiation. Example in zoology, the medium brown finch Geospiza fortis has a beak adapted for cracking hard seeds, while the cactus finch Geospiza scandens has a longer and more pointed beak suited for probing flowers and eating cactus nectar. These variations in beak morphology among Darwin's finches demonstrate how natural selection can drive adaptive changes within a population. Pesticide resistance in insects. Insects, such as mosquitoes and agricultural pests, can develop resistance to pesticides through genetic mutations or gene transfer. Individuals with resistant traits have survival advantage when exposed to pesticides, allowing them to survive and reproduce. Consequently, the resistant traits become more prevalent in the population over time. Example in zoology, the Colorado potato beetle Leptinotarsivis emelinata is an agricultural pest that has developed resistance to various insecticides. Continuous exposure to these chemicals has led to the emergence of resistant populations, posing challenges for pest control in potato crops. These examples demonstrate how evolution in action can be observed in real-world scenarios, providing evidence for the ongoing process of genetic change and adaptation. Through natural selection, genetic variations that confer advantages in specific environments become more prevalent in populations over time. The study of evolution in action allows scientists to understand how species respond and adapt to changing conditions, providing insights into imperfection of evolution. ChatGPT. While the theory of evolution is widely accepted in the scientific community and supported by substantial evidence, it is important to acknowledge that evolution is not a perfect process. There are certain imperfections and limitations associated with evolution. Here are some key points regarding the imperfections of evolution. Suboptimal design. One criticism of evolution is the presence of suboptimal or seemingly inefficient features in organisms. Some argue that if evolution is an adaptive process driven by natural selection, why are there instances of imperfect or less than ideal designs? Examples include the inverted retina in the eyes of vertebrates, which can lead to reduced visual acuity, or the recurrent laryngeal nerve in giraffes, which takes a lengthy detour through the chest before reaching the larynx. Constraints and trade-offs. Evolution often works within certain constraints and trade-offs due to the existing genetic variations and developmental pathways. This can lead to compromises in the evolutionary process. For instance, the trade-off between speed and accuracy in DNA replication can result in mutations and genetic errors. Similarly, the size and shape of certain structures may be limited by developmental constraints. Vestigial structures. Vestigial structures are remnants of ancestral features that have lost their original function in the course of evolution. These structures are considered to be evolutionary leftovers. Examples include the appendix in humans, which has a reduced role in digestion, and the wings of flightless birds like the ostrich. These vestigial structures are often cited as evidence of imperfect or inefficient design. Genetic drift. While natural selection is a significant driving force of evolution, genetic drift can also influence the genetic composition of populations. Genetic drift refers to random fluctuations in allele frequencies due to chance events. In small populations, genetic drift can have a more significant impact, leading to the loss or fixation of alleles without necessarily conferring adaptive benefits. Evolutionary lag. Evolution does not occur instantaneously but takes place over extended periods of time. As a result, organisms may experience a lag in adapting to new environmental changes. This lag can lead to mismatches between an organism's traits and its current environment. For example, human bodies are adapted to hunter-gatherer lifestyle, but the rapid changes in modern society and technology have created new challenges for human health and well-being. Extinction. Evolutionary processes also involve the extinction of species. While new species emerge through speciation, others become extinct due to various factors such as environmental changes, competition, or inability to adapt. The occurrence of extinction implies that not all lineages or species are successful in the long run, highlighting the imperfections and uncertainties associated with the evolutionary process. It is important to note that these imperfections and limitations do not undermine the overall validity and robustness of the theory of evolution. Evolutionary biology continues to advance our understanding of the natural world and provides crucial insights into the diversity of life. The imperfections observed can often be attributed to the complex and dynamic nature of evolutionary processes, as well as the diverse range of environmental factors that influence them. Imperfection of evolution in detail with respect to evolution with examples of zoology in detail. ChatGPT. The theory of evolution, while widely accepted as the prevailing explanation for the diversity of life, is not without imperfections. Here, we delve into some of the imperfections of evolution and provide examples from the field of zoology. Suboptimal adaptations. Evolution is driven by natural selection, favoring traits that enhance an organism's survival and reproductive success. However, not all adaptations are perfect. For instance, the panda's thumb is an example of an imperfect adaptation. The thumb-like extension of its wrist bone aids in grasping bamboo, its primary food source, but it is less dexterous than a true opposable thumb. Coevolutionary arms race. Species often engage in coevolutionary interactions, such as predator-prey relationships or host-parasite interactions. These interactions involve a constant battle for survival, where both sides continuously evolve and counter-adapt to gain an advantage. Imperfections can arise when one species evolves a new trait, and the other species must adapt in response. This leads to a continuous arms race, rather than achieving perfect adaptation. Genetic constraints. Evolution works with the genetic variation present within a population. Certain genetic constraints can limit the range of evolutionary possibilities. For example, the genetic code is universal across all living organisms, meaning certain changes in DNA sequences may have deleterious effects. This constraint can hinder the development of novel traits and limit the potential outcomes of evolution. Evolutionary legacy. Organisms carry a history of their evolutionary past, which can impose constraints on their current form and function. Some structures may have persisted despite being less than optimal due to their ancestral legacy. For example, the wings of flightless birds, like the kiwi, were reduced in non-functional remnants of their flying ancestors. Evolutionary lag. Evolution is a gradual process that occurs over generations, and it takes time for organisms to adapt to new environmental conditions. In rapidly changing environments, such as those impacted by human activities, organisms may face challenges in keeping up with the pace of change. This lag can result in imperfect adaptations or mismatches between an organism's traits and its environment. Genetic load. Over time, genetic mutations accumulate in populations, some of which can be deleterious. These mutations may not be immediately eliminated from the population due to factors such as genetic drift or low selection pressure. As a result, populations may carry a genetic load of deleterious mutations, which can have negative effects on individual fitness. Imperfect predictability. While evolution is a scientific theory backed by extensive evidence, it is not always predictable at the level of specific outcomes. Evolutionary processes are influenced by numerous factors, including chance events, genetic interactions, and environmental changes. As a result, the specific path and outcomes of evolution can be difficult to predict accurately. It is important to note that these imperfections do not invalidate the theory of evolution. Rather, they reflect the complexities and constraints inherent in the evolutionary process. Imperfections in evolution highlight the ongoing nature of scientific inquiry and the dynamic nature of life on Earth. Through continued research and investigation, scientists strive to gain a deeper understanding of the intricacies of evolution and the imperfections that arise from its complex mechanisms. Coevolution, microevolution, in detail with respect to evolution with examples of zoology in detail. ChatGPT. Coevolution refers to the reciprocal evolutionary changes that occur between two or more species as a result of their interactions. These interactions can be cooperative, such as mutualistic relationships, or antagonistic, such as predator-prey or host-parasite relationships. Coevolution operates at the level of microevolution, involving genetic changes within populations over relatively short periods of time. Here, we explore coevolution in detail with examples from zoology. Predator-prey coevolution. Predator-prey interactions often drive coevolutionary dynamics. Predators exert selection pressure on their prey, favoring individuals with traits that enhance their ability to escape predation. Prey, in turn, develop defensive adaptations to avoid being captured. Classic examples include the arms race between cheetahs, predators, and gazelles prey. Cheetahs have evolved incredible speed and agility to catch fast-running gazelles, while gazelles have developed swift movements and keen senses to evade predation. Host-parasite coevolution. Host-parasite interactions provide another compelling example of coevolution. Parasites, such as ticks, fleas, and lice, rely on their hosts for food and reproduction. Hosts, on the other hand, evolve defenses against these parasites to reduce their negative impact. This results in a constant back-and-forth evolution of resistance and countermeasures. A well-known example is the coevolution between birds and their brood parasites, like cuckoos. Cuckoos lay their eggs in the nests of other bird species, and host birds have evolved strategies to recognize and reject foreign eggs. Mutualistic company evolution. Mutualistic interactions, where two species benefit from their association, also undergo coevolution. One example is the relationship between flowering plants and their pollinators. Plants produce nectar to attract pollinators, such as bees or hummingbirds, which inadvertently transfer pollen from one flower to another. As plants evolve features like colorful petals or sweet nectar to attract specific pollinators, the pollinators, in turn, develop specialized mouthparts or behaviors to efficiently access the rewards. This coevolutionary process ensures the mutual benefit of both parties. Plant-herbivore coevolution. Herbivores consume plants as a food source, and plants have evolved various mechanisms to defend themselves. This leads to coevolutionary interactions between herbivores and plants. For example, some plants have evolved chemical compounds, such as toxins or bitter-tasting substances, to deter herbivores. In response, herbivores may develop specialized detoxification enzymes or feeding adaptations to overcome these defenses. The ongoing coevolution between milkweed plants and monarch butterflies is a well-known example, where monarchs have evolved the ability to feed on milkweed plants despite their toxic chemicals. Co-evolutionary processes occur throughout the animal kingdom, shaping the traits and adaptations of species involved. These interactions drive genetic changes within populations, allowing organisms to better exploit resources, avoid predators, or enhance reproductive success. Co-evolution provides a dynamic and intricate pattern of reciprocal adaptations that contribute to the biodiversity and ecological relationships observed in the natural world. Macroevolution, in detail with respect to evolution with examples of zoology in detail. ChatGPT. Macroevolution refers to large-scale evolutionary patterns and processes that occur over extended periods of time, resulting in the formation of new species, changes in major lineages, and the diversification of life forms. It involves the study of evolutionary changes that are above the species level. Here, we explore macroevolution in detail with examples from zoology. Speciation. Speciation is the process by which new species arise from existing ones. It occurs when populations become reproductively isolated and diverge genetically to the point where they can no longer interbreed. This can happen through various mechanisms, such as geographic isolation, allopatric speciation, or within the same geographic range, sympatric speciation. For example, Darwin's finches in the Galapagos Islands provide a classic example of adaptive radiation, where a single ancestral species diversified into multiple species with different beak shapes and feeding habits. Extinction. Extinction is a crucial aspect of macroevolution as it shapes the diversity and composition of life on Earth. Mass extinctions, such as the Cretaceous-Paleogene extinction that wiped out the dinosaurs, have had profound impacts on the course of evolution. Extinctions create opportunities for surviving species to adapt to vacant ecological niches, leading to the subsequent diversification and radiation of new lineages. For example, after the extinction of dinosaurs, mammals experienced a rapid diversification and eventually became the dominant group of land vertebrates. Adaptive radiations. Adaptive radiations occur when a single ancestral species diversifies into a wide array of ecological niches, often in response to the availability of new habitats or ecological opportunities. This results in the rapid evolution of numerous new species with distinct adaptations. An example of adaptive radiation is the diversification of mammals following the extinction of dinosaurs. Mammals filled various ecological roles and evolved characteristics suited for different environments, leading to the evolution of primates, rodents, carnivores, and many other mammalian groups. Convergent evolution. Convergent evolution is the process by which unrelated species independently evolve similar traits or adaptations in response to similar selective pressures. This phenomenon is often observed in different lineages occupying similar ecological niches. For example, dolphins and sharks have evolved similar streamlined body shapes and fin-like structures for efficient swimming, despite their distant evolutionary relationships. This convergence of traits is a result of adapting to similar aquatic environments and lifestyles. Co-evolutionary patterns. Macroevolution also encompasses co-evolutionary patterns and interactions between different species over long periods. These interactions can result in reciprocal evolutionary changes, such as predator-prey relationships, mutualistic associations, or host-parasite interactions. The co-evolutionary arms race between predators and prey, as well as the co-evolution between plants and their pollinators, are examples of macroevolutionary processes that shape the diversification and adaptation of species. Macroevolutionary processes are essential for understanding the broader patterns of life's history and the diversity of organisms we see today. By macroevolution, scientists gain insights into the origin of major groups, the mechanisms that drive speciation and extinction, and the complex interactions between organisms and ecosystems. It provides a comprehensive perspective on the long-term evolutionary changes that have shaped the biodiversity and complexity of life on Earth. Convergent evolution. Homoplasy, divergent, parallel evolution. In detail with respect to evolution with examples of zoology in detail. ChatGPT. Convergent evolution and divergent evolution are two contrasting patterns of evolution that occur in different species. Let's explore them in detail with examples from zoology. Convergent evolution, homoplasy. Convergent evolution refers to the independent evolution of similar traits or adaptations in unrelated species that face similar environmental challenges. It occurs when different organisms, often from different lineages, develop analogous structures or functions to adapt to similar ecological niches or selective pressures. The similarity in traits is not due to common ancestry but rather a result of adapting to similar environmental conditions. Examples of convergent evolution in zoology include wings, birds, bats, and insects all have wings and the ability to fly, but they evolve this trait independently. Their wings have different structures and are composed of different materials, feathers, membranes, or chitin, yet they serve a similar purpose of enabling flight. Echolocation. Bats and dolphins both use echolocation to navigate and locate prey in their environments. They have evolved the ability to emit high-frequency sounds and interpret the echoes to determine the location of objects. Despite their distinct evolutionary lineages, they have independently evolved this sensory adaptation for similar purposes. Camouflage. Various species from different taxonomic groups have evolved similar camouflage patterns to blend into their environments. For example, both the Arctic fox Vulpes lagopus and the snowshoe hare Lepus americanus exhibit a white coloration during winter in snowy habitats. This convergence in coloration helps them evade predators and remain hidden in their respective environments. Divergent evolution, parallel evolution. Divergent evolution refers to the process by which closely related species or populations evolve different traits or adaptations over time. It occurs when species or populations diverge from a common ancestor and undergo independent evolutionary paths. Divergent evolution can lead to the formation of new species or the development of distinct traits in different populations. Examples of divergent evolution in zoology include Darwin's finches. The finches on the Galapagos Islands, famously studied by Charles Darwin, provide a classic example of divergent evolution. Different species of finches evolved distinct beak shapes and sizes, enabling them to specialize in different food sources available on the islands. This divergence in beak morphology was driven by adaptation to different ecological niches and resulted in the formation of new species. Anoles lizards. Anoles lizards in the Caribbean islands have evolved different body sizes and limb lengths depending on the habitat they occupy. In open habitats, they have longer limbs for faster running, while in densely vegetated habitats, they have shorter limbs for better maneuverability. This divergence in morphology allows them to exploit different microhabitats within their range. Cichlid fish. Cichlid fish in African lakes, such as Lake Victoria, have undergone rapid divergent evolution, leading to the formation of numerous species with distinct morphological and behavioral adaptations. Different species have evolved variations in jaw shape, coloration, and feeding habits, allowing them to occupy different ecological niches and reduce competition. Convergent evolution and divergent evolution are both important processes that shape the diversity and complexity of life on Earth. They highlight the remarkable capacity of organisms to adapt to their environments and provide evidence for the influence of natural selection and environmental pressures on evolutionary outcomes. By studying these patterns, scientists gain insights into the underlying mechanisms of adaptation and the evolutionary history of organisms in different ecosystems. Tempo of evolution in detail with respect to evolution with examples of zoology in detail. ChatGPT. The tempo of evolution refers to the rate or speed at which evolutionary changes occur in a given lineage or population over time. It can vary widely depending on several factors, including the intensity of natural selection, genetic variation, and environmental conditions. Let's explore the tempo of evolution in more detail with examples from zoology. Gradualism. Gradualism is a concept that suggests evolutionary changes occur gradually and steadily over long periods of time. In this mode of evolution, species undergo small, incremental changes over generations, leading to the accumulation of significant differences over extended periods. Examples of gradualism in zoology include horse evolution. The evolution of the horse is often cited as an example of gradualism. Fossil records show a series of intermediate forms from early horse-like ancestors to the modern-day horse. Each fossil reveals small changes in size, dental structure, and limb adaptations, illustrating the gradual transition from four-toed ancestors to the single-toed modern horse. Snail shell coiling. Some species of snails exhibit coiled shells with varying degrees of spiral winding. Fossil records show a gradual change in shell coiling patterns over time, with intermediate forms demonstrating a step-by-step transition from one coiling direction to another. Punctuated equilibrium. Punctuated equilibrium suggests that evolutionary change occurs rapidly in brief periods of time, punctuated by long periods of relative stability. This mode of evolution proposes that species experience little to no change for most of their existence, with significant modifications occurring during periods of rapid speciation or environmental changes. Examples of punctuated equilibrium in zoology include trilobite evolution. Trilobites were a diverse group of marine arthropods that existed for over 270 million years. Fossil records reveal long periods of morphological stasis, where trilobite species remained relatively unchanged, followed by sudden bursts of diversification and speciation during episodes of environmental upheaval or geological events. Cichlid fish radiations. Cichlid fish in African lakes, such as Lake Malawi and Lake Tanganyika, underwent rapid diversification and speciation in relatively short periods of time. Environmental changes, such as changes in lake water chemistry or habitat availability, triggered intense adaptive radiations, leading to the rapid emergence of numerous distinct cichlid species. Adaptive radiation. Adaptive radiation occurs when a single ancestral species gives rise to multiple descendant species that rapidly diversify and occupy various ecological niches. It often happens in environments with abundant available resources and few existing competitors. Examples of adaptive radiation in zoology include Darwin's finches. The finches in the Galapagos Islands provide a classic example of adaptive radiation. A single ancestral finch species colonized the islands and, over time, diversified into several distinct species with different beak shapes and feeding habits. Species adapted to a specific food source, such as seeds, insects, or cactus flowers, allowing them to occupy different ecological niches. Hawaiian honeycreepers. The Hawaiian Islands are home to a remarkable adaptive radiation of honeycreepers. These birds evolved from a common ancestor and radiated into numerous species, each with unique beak shapes and feeding strategies adapted to specific ecological niches, such as nectar feeding, seed cracking, or insect probing. The tempo of evolution can vary significantly depending on the circumstances and evolutionary pressures faced by organisms. Some lineages may experience slow and steady changes over long periods, while others may undergo rapid diversification in response to environmental or ecological opportunities. By studying the tempo of evolution, scientists gain insights into the pace and patterns of evolutionary change and how species adapt and diversify over time. Gradualism versus punctuated equilibrium. In detail with respect to evolution with examples of zoology in detail. ChatGPT. Gradualism and punctuated equilibrium are contrasting models that describe different patterns of evolutionary change over time. Let's explore the concepts of gradualism and punctuated equilibrium in more detail, along with examples from zoology. Gradualism. Gradualism proposes that evolutionary change occurs gradually and steadily over long periods of time. According to this model, species undergo small, incremental changes in their traits and characteristics, leading to the accumulation of significant differences over extended periods. Key points about gradualism include continuous change. Gradualism suggests that species evolve through a continuous process of small, adaptive changes in response to selective pressures. This gradual accumulation of changes results in the transformation of populations over time. Transitional fossils. Gradualism predicts the existence of transitional fossils, which are intermediate forms that exhibit traits intermediate between ancestral and descendant species. These fossils provide evidence for the gradual transformation of species. Example: the evolution of the horse is often cited as an example of gradualism. Fossil records of the horse lineage show a series of intermediate forms with gradual changes in size, dental structure, and limb adaptations. This sequence of fossils demonstrates the gradual transition from small, multi-toed ancestors to the larger, single-toed modern horses. Punctuated equilibrium. Punctuated equilibrium proposes that evolutionary change occurs rapidly in brief periods of time, punctuated by long periods of relative stability. According to this model, species experience long periods of stasis with little to no change, followed by relatively short bursts of rapid speciation or significant morphological change. Key points about punctuated equilibrium include periods of stasis. Punctuated equilibrium suggests that most species experience little evolutionary change during their existence. They remain relatively stable in terms of morphology and adaptation for long periods, as long as their environment remains stable. Rapid change. Punctuated equilibrium proposes that evolutionary change occurs during relatively short periods of time when species encounter new environments or face sudden selective pressures. These events trigger intense periods of speciation and diversification. Example: the fossil record of trilobites provides evidence for punctuated equilibrium. Trilobites were marine arthropods that existed for millions of years. Fossil evidence shows long periods of morphological stasis, where trilobite species remained relatively unchanged, followed by sudden bursts of diversification and speciation during episodes of environmental upheaval or geological events. While gradualism and punctuated equilibrium represent contrasting models, they are not mutually exclusive. Evolutionary change can occur through a combination of gradual accumulation of small changes and rapid episodes of speciation or significant shifts in morphology. The relative importance of these patterns may vary depending on the species, the selective pressures they face, and the timescale of observation. Both models contribute to our understanding of the diversity of life and the dynamic nature of evolutionary processes. More points to halves. ChatGPT. Certainly. Here are some additional points regarding gradualism and punctuated equilibrium in the context of evolution. Gradualism. Adaptive change. Gradualism emphasizes that evolutionary change is driven by natural selection acting on small variations within populations. Beneficial traits increase in frequency over time through the accumulation of small, adaptive changes. Transitional forms. Gradualism predicts the existence of numerous transitional forms in the fossil record, representing intermediate stages between ancestral and descendant species. These transitional forms provide evidence for the gradual transformation of lineages. Selection pressure. Gradualism suggests that the environment exerts a continuous selection pressure, favoring individuals with traits that confer a reproductive advantage. Over generations, this leads to the gradual spread of advantageous traits throughout a population. Punctuated equilibrium. Rapid speciation. Punctuated equilibrium proposes that speciation events can occur relatively quickly, leading to the emergence of new species in a year. Gradualism versus punctuated equilibrium. In detail with respect to evolution with examples of zoology in detail. ChatGPT. Gradualism and punctuated equilibrium are contrasting models that describe different patterns of evolutionary change over time. Let's explore the concepts of gradualism and punctuated equilibrium in more detail, along with examples from zoology. Gradualism. Gradualism proposes that evolutionary change occurs gradually and steadily over long periods of time. According to this model, species undergo small, incremental changes in their traits and characteristics, leading to the accumulation of significant differences over extended periods. Key points about gradualism include continuous change. Gradualism suggests that species evolve through a continuous process of small, adaptive changes in response to selective pressures. This gradual accumulation of changes results in the transformation of populations over time. Transfossils. Gradualism predicts the existence of transitional fossils, which are intermediate forms that exhibit traits intermediate between ancestral and descendant species. These fossils provide evidence for the gradual transformation of species. Example: the evolution of the horse is often cited as an example of gradualism. Fossil records of the horse lineage show a series of intermediate forms with gradual changes in size, dental structure, and limb adaptations. This sequence of fossils demonstrates the gradual transition from small, multi-toed ancestors to the larger, single-toed modern horses. Punctuated equilibrium. Punctuated equilibrium proposes that evolutionary change occurs rapidly in brief periods of time, punctuated by long periods of relative stability. According to this model, species experience long periods of stasis with little to no change, followed by relatively short bursts of rapid speciation or significant morphological change. Key points about punctuated equilibrium include periods of stasis. Punctuated equilibrium suggests that most species experience little evolutionary change during their existence. They remain relatively stable in terms of morphology and adaptation for long periods, as long as their environment remains stable. Rapid change. Punctuated equilibrium proposes that evolutionary change occurs during relatively short periods of time when species encounter new environments or face sudden selective pressures. These events trigger intense periods of speciation and diversification. Example: the fossil record of trilobites provides evidence for punctuated equilibrium. Trilobites were marine arthropods that existed for millions of years. Fossil evidence shows long periods of morphological stasis, where trilobite species remained relatively unchanged, followed by sudden bursts of diversification and speciation during episodes of environmental upheaval or geological events. While gradualism and punctuated equilibrium represent contrasting models, they are not mutually exclusive. Evolutionary change can occur through a combination of gradual accumulation of small changes and rapid episodes of speciation or significant shifts in morphology. The relative importance of these patterns may vary depending on the species, the selective pressures they face, and the timescale of observation. Both models contribute to our understanding of the diversity of life and the dynamic nature of evolutionary processes. More points to halves. ChatGPT. Certainly. Here are some additional points regarding gradualism and punctuated equilibrium in the context of evolution. Gradualism. Adaptive change. Gradualism emphasizes that evolutionary change is driven by natural selection acting on small variations within populations. Beneficial traits increase in frequency over time through the accumulation of small, adaptive changes. Transitional forms. Gradualism predicts the existence of numerous transitional forms in the fossil record, representing intermediate stages between ancestral and descendant species. These transitional forms provide evidence for the gradual transformation of lineages. Selection pressure. Gradualism suggests that the environment exerts a continuous selection pressure, favoring individuals with traits that confer a reproductive advantage. Over generations, this leads to the gradual spread of advantageous traits throughout a population. Punctuated equilibrium. Rapid speciation. Punctuated equilibrium proposes that speciation events can occur relatively quickly, leading to the emergence of new species in a short period. These speciation events often coincide with changes in environmental conditions or the colonization of new habitats. Genetic isolation. Punctuated equilibrium suggests that during periods of stability, populations may become genetically isolated from one another, reducing gene flow. This isolation allows for the accumulation of genetic changes within each population, leading to distinct evolutionary trajectories. Morphological change. Punctuated equilibrium highlights that periods of rapid evolution can result in significant morphological changes. This can include the development of new traits or the loss of ancestral traits, leading to the formation of distinct forms or ecological adaptations. Stabilizing selection. Punctuated equilibrium suggests that once a species reaches a stable and successful form, natural selection may act to maintain its characteristics over long periods of stasis. This occurs when a species is well adapted to its environment and experiences minimal selective pressures. It's important to note that the debate between gradualism and punctuated equilibrium is not about whether one is entirely correct and the other is incorrect. Instead, it reflects different perspectives on the tempo and pattern of evolutionary change. Both models contribute to our understanding of how species arise and diversify over time, and the relative importance of each process may vary depending on the specific circumstances and taxa under consideration. Anagenesis versus cladogenesis. In detail with respect to evolution with examples of zoology and detail ChatGPT. Certainly. Here are some points regarding anagenesis and cladogenesis in the context of evolution. Anagenesis. Definition. Anagenesis refers to the gradual evolutionary change within a single lineage over time. It involves the accumulation of genetic and phenotypic changes in a population, resulting in the transformation of the ancestral form into a new form. Trait evolution. Anagenesis often leads to the gradual transformation of traits within a species. This can include changes in morphology, physiology, behavior, or other characteristics that enhance the adaptation and survival of individuals in their environment. Continuous lineage. Anagenesis occurs within a continuous lineage, with successive generations exhibiting gradual changes in traits. The ancestral form is replaced by a descendant form through the accumulation of small, incremental changes. Lack of speciation. Anagenesis does not involve the splitting of lineages or the formation of new species. Instead, it represents the transformation of a single lineage over time. Example: the evolution of the horse lineage is often cited as an example of anagenesis. Fossil evidence shows a gradual series of changes in horse species over millions of years, including size increase, development of a single toe to a hoof, and changes in dental morphology. Cladogenesis. Definition. Cladogenesis refers to the branching of a single lineage into two or more distinct lineages, resulting in the formation of new species. It involves the splitting of a population into separate evolutionary paths, leading to the divergence of traits and the formation of distinct lineages. Speciation event. Cladogenesis occurs when a population becomes reproductively isolated, preventing gene flow between subpopulations. This can happen through various mechanisms such as geographic isolation, ecological specialization, or genetic divergence. Divergence of traits. Cladogenesis leads to the divergence of traits between the newly formed lineages. Each lineage may experience different selective pressures and environmental conditions, resulting in the accumulation of distinct genetic and phenotypic characteristics. Increase in biodiversity. Cladogenesis is a key process driving the increase in biodiversity over evolutionary time. By generating new species, it contributes to the expansion and diversification of life forms on Earth. Example: the adaptive radiation of Darwin's finches in the Galapagos Islands is a classic example of cladogenesis. The finches diversified from a common ancestor into multiple species with different beak sizes and shapes, allowing them to exploit different food resources on different islands. It's important to note that anagenesis and cladogenesis are not mutually exclusive processes. Evolutionary change can occur both within lineages, anagenesis, and through the splitting of lineages, cladogenesis. These processes often work in combination to shape the patterns of biodiversity and species evolution. More points 5/5. ChatGPT. Certainly. Here are some additional points regarding anagenesis and cladogenesis in the context of evolution. Anagenesis. Gradual accumulation. Anagenesis involves the accumulation of genetic and phenotypic changes over long periods of time. The rate of change can vary, with some lineages experiencing relatively rapid transformations while others undergo more gradual shifts. Environmental influence. Anagenesis is influenced by environmental factors, such as changes in climate, habitat, or resource availability. These factors can drive natural selection and shape the direction of evolutionary change within a lineage. Genetic variation. Anagenesis relies on genetic variation within a population. Mutations, genetic recombination, and other mechanisms contribute to the introduction of new genetic variants, providing the raw material for evolutionary change. Cladogenesis. Reproductive isolation. Cladogenesis occurs when reproductive isolation occurs between subpopulations. This can be due to various factors, including geographic barriers, changes in mating behaviors, or genetic incompatibilities. The isolation prevents gene flow and allows for independent evolutionary trajectories. Adaptive radiation. Cladogenesis often leads to adaptive radiation, where the newly formed lineages occupy different ecological niches and adapt to diverse environments. This can result in the rapid diversification of species and the occupation of various ecological roles. Speciation modes. Cladogenesis can occur through different speciation modes. These include allopatric speciation, where geographic barriers separate populations, sympatric speciation, where reproductive isolation arises within the same geographic area, and parapatric speciation, where populations diverge while maintaining limited gene flow. Extinction and persistence. Cladogenesis can lead to the extinction of ancestral lineages, as well as the persistence of some lineages alongside the newly formed ones. This dynamic interplay between extinction and persistence contributes to the overall patterns of biodiversity. It's important to understand that anagenesis and cladogenesis are concepts used to describe different modes of evolutionary change. In reality, evolution is a complex and dynamic process involving a combination of both gradual changes within lineages and the splitting of lineages into new branches. These processes interact with various ecological, genetic, and environmental factors to shape the diversity of life on Earth. Molecular evolutions in detail with respect to evolution with examples of zoology in detail. ChatGPT. Molecular evolution is the study of changes in the genetic material, DNA or RNA, of organisms over time. It provides insights into the patterns and processes of evolution at the molecular level. Here are some key points regarding molecular evolution with respect to evolution, along with examples from zoology. Molecular clock. The concept of a molecular clock suggests that the rate of molecular change is relatively constant over time. This can be used to estimate the divergence times between different species or lineages. For example, by comparing the DNA sequences of different primates, researchers have estimated the divergence between humans and chimpanzees to have occurred around 6 to 7 million years ago. Genetic variation and adaptation. Molecular evolution allows us to understand how genetic variation contributes to adaptation and evolutionary change. Genetic variations, such as single nucleotide polymorphisms, SNPs, or structural variations, can confer selective advantages or disadvantages in different environments. For instance, in the study of Darwin's finches, researchers have identified specific genes associated with beak morphology, which is an adaptation to different diets. Gene duplication and divergence. Gene duplication events can provide a source of new genetic material for evolution. When a gene is duplicated, one copy can continue to perform its original function, while the other copy is free to accumulate mutations and undergo functional divergence. This process can lead to the evolution of new genes and functions. An example is the opsin genes responsible for color vision in primates, which have undergone several duplication events and subsequent divergence to enable color vision in different light conditions. Molecular phylogenetics. Molecular data, such as DNA sequences, can be used to reconstruct the evolutionary relationships among species or populations. Phylogenetic trees based on molecular data provide insights into the evolutionary history and relatedness of organisms. For example, molecular phylogenetics has helped to resolve the relationships among different bird species, uncovering their evolutionary history and patterns of diversification. Molecular adaptations. Molecular evolution also reveals adaptations at the molecular level. Certain genes or regions of the genome may show signatures of positive selection, indicating they have undergone adaptive changes in response to environmental pressures. For instance, in the study of antifreeze proteins in Arctic fish, researchers have found evidence of positive selection acting on specific amino acid positions, allowing the fish to survive in freezing waters. Molecular convergence. Molecular evolution can also uncover instances of convergent evolution, where different species independently evolve similar traits or genetic changes in response to similar environmental challenges. For example, the evolution of echolocation in bats and dolphins has involved convergent changes in genes related to auditory perception. Molecular evolution provides a powerful tool for understanding the mechanisms and patterns of evolution. By analyzing genetic sequences and studying molecular changes over time, researchers can gain insights into the evolutionary history, genetic adaptations, and diversification of organisms in the animal kingdom. Neutral theory of molecular evolution. In detail with respect to evolution with examples of zoology in detail. ChatGPT. The neutral theory of molecular evolution is a concept proposed by Japanese scientist Moto Kimura in the 1960s. It suggests that a significant portion of genetic variation observed at the molecular level is due to the accumulation of neutral mutations that do not affect an organism's fitness or phenotype. Here are some key points regarding the neutral theory of molecular evolution with respect to evolution, along with examples from zoology. Molecular clock. The neutral theory of molecular evolution assumes that the rate of neutral mutation accumulation is relatively constant over time. This implies that the genetic differences between species or lineages are primarily a result of the accumulation of neutral mutations, rather than adaptive changes. By